Adventures with Vegetable Pests. The emphasis is actually going to be more on the semiochemicals and behavior, but we'll see how it goes. These are some of my stars. Beetles and true bugs, and there's even a hymenoptene there in the corner. Here are the common names and a few scientific names of the beetles, bugs, and there's even a lepidoptera in there. So a large part of my talk's going to be concerning male-produced aggregation pheromones, and those are really common in beetles, including chrysomelids, leaf beetles, longhorn beetles, and buprestids, as well as several families of true bugs, including Panatomidae, the stink bugs, and Coreidae, the leaf-footed bugs. In this case, the male produces a pheromone, and it's attractive to both males and females. And interestingly enough, we're tested to nymphs. There's some potential for use in management. Not many of them are known, but where they're known and produced in enough quantity to test in the field, they're pretty powerful tools. But there's a lot of steps between that discovery and any application to management of vegetable pests. And here are some target species I've listed. They're probably familiar to some of you, maybe too familiar in some cases, like the brown marmorated stink bug, and perhaps if you've grown potatoes, the Colorado potato beetle. So each species has some different ways it operates, and like the two there are thinking, there's always a wrinkle. I'm going to start out with some work we did with brown marmorated stink bug. Jeff Aldrich was in my lab and agreed to be the main stink bug person for 5 years, and then he retired after a year. So it came to me to work on the brown marmorated stink bug, which was in great invasive waves in the mid-Atlantic at the time. So this is a little chemistry here showing the isomers of the two stink bugs, and I just introduce it in the way of showing that there are some commonalities among different species, but there's also some subtle differences. And in this case, we really got a twofer, because with the work of Ashad Krimian, our research chemist in the group, he was able to identify each and every isomer of the compound you see here, which was dubbed mergantiol, and 2 of them are harlequin bug pheromone, and 2 of them with one in common are the brown marmorated stink bug pheromone. Interestingly, with the brown marmorated stink bug, the first attractant that was well known was actually the pheromone of another stink bug named Plautia stahli from Asia, where it co-occurs with brown marmorated stink bug. And its pheromone, methodecatrienoate, was attractive to brown marmorated, but only in the fall. So not very useful if you're trying to grow apples and you want to know what's coming at me, because you're only going to know later after your apples are perhaps damaged by brown marmorated. So knowing that, and with the discovery of the pheromone, the true pheromone of the brown marmorated stink bug, we tested the combination, as you can see from the green bars there. When we add the 2 attractants together, we often got a synergistic attractive effect. And this was very useful in terms of applied monitoring, because all we had to do was say, use both lures all season long. And the data backed that up for every single month. And as I mentioned before, it's also attractive to the nymphs. So, and the season-long differences aren't very different between the adults and the nymphs. And the males and females are, as the term aggregation pheromone implies, equally attractive. So we did some rather subtle work with the isomers that Ashok Krimian came up with. And something interesting here, maybe mainly for scientific purposes, the females, as indicated in blue here, are much more attracted to the natural blend, and the males aren't too fussy. Well, in a way, that makes sense. I think in evolutionary terms, if you think the females want a male, a mate in his prime that produces the exact blend, and the males just want to get to where the females are. So another interesting thing that we observe with a lot of aggregation pheromones is that there's no upper limit. So if you know about Lepidoptera sex pheromones that are the best studied group of pheromones, if you put out a very high dose, the males that are going to where they think a female is will not go to the source if it's a very high dose. There's a limit there. They, I guess you could say they smell a rat. They think, what's going on here? There's no female that's putting out that much. However, with this group of male-produced aggregation pheromones, we don't see that. And that's, in a way, an advantage when you think about management and possible applications to attracting in large groups, which Tracy Leskey's group is pictured here, sorting out literally thousands of brown marmorated stink bugs, attracted to as much as a gram of the pheromone, along with the synergist, the Plautia stali pheromone that I mentioned. So this cross-species attraction in this case was very interesting. We don't really know why that is. There's some proposed reasons. Maybe it's a food signal. They're both polyphagous species, meaning they feed on a lot of different plants. They're eavesdropping on one another's signals. This signal is most, it's actually possibly an overwintering site signal. In Asia, they overwinter in common sites in hollow trees and holes in the rocks, and like in North America, in a very irritating way in people's houses. So Jeff also had the idea that maybe if they aggregate together, that passively protects them from natural enemies. Not a tested hypothesis yet. All right, on to harlequin bug, which, as I mentioned, the commercial pheromone now is a mix of 8 isomers that include all 3 that you see here. And with harlequin bug, we were interested to see how that worked, and getting rid of for now of the brown marmorated, combining with the harlequin bug as a mustard family specialist. It's a big pest on coal crops, and the idea that we could combine that with the isothiocyanates, which are mustard oils, and attracted to many mustard family herbivores. So this was some of the harlequin bug pheromone identification, just a quick expedition into the chemistry here. And the 2 isomers that are the true male pheromone overlap with the 4 isomers that Dr. Krimian produced. And you can see there in the middle how they perfectly synchrony on gas chromatograph output. And as with the brown marmorated, the females are fussy about the ratio, but they're all attracted to various combinations of the 2 components of the pheromone. And the males are less fussy, as you can see the contrast where it says, yes, that's a significant effect, the natural ratio versus the off ratio. The females are sensitive to that. So this was a very simple and low-tech way that we looked at choices that the harlequin bugs could make in the field. A collared plant with a hat that had a pheromone lure in it, and maybe it's a blank and maybe it's a different ratio depending on what we're testing. And so when we tested, for instance, blank versus collar plus pheromone, you can see the top bar, there's a big effect. And it's basically almost none to the collared and a lot to the collared with the pheromone. Similarly, if you test a host versus a non-host, and the non-host in this case, soybean, a big effect. We wanted to see if we could fool the harlequin bug. And when we put both pheromones, there is some attraction to the soybean, but when we put only the pheromone on the soybean, the bottom set of bars there, you can see we have fooled the harlequin bug. And if you know what a harlequin is, they're all about fooling. So we were also interested in this phenomenon, which we always have to keep in mind with attraction and arrestment. So a bug can be attracted to a plant, but if it leaves soon afterwards, you might not be able to observe that. So what we're looking at here is a situation where on the top was the example I showed where we just had a collard and then a soybean plus pheromone. And we observed these twice a day. So during that time, for all we knew, bugs could arrive at the soybean having smelled the pheromone and then leave. And in fact, that was true because when we poisoned the plants in such a way that when the bugs came and took a very small sample, they would die underneath the plant. You can see the bars, the second bar there underneath, those are both treated. And instead of getting a 3.8 times effect, we got a 269 times effect. So 269 times more bugs were arriving at the soybean with the pheromone. So this is very interesting. You can see the bottom here. If we hadn't poisoned the plants and just observed them twice a day, we would think, oh, there's no effect of putting pheromone plus benzyl isocyanate on it. But when we used the poison plants, we saw that there was a 25x difference. So this is very important in terms of trap cropping or any type of manipulation to distinguish between arrival and departure, and in the behavioral attraction versus does it stay there, arrestment. So just a brief kind of excursion into applying some of this knowledge. We have a discovery. First component might be, hey, there's a male bug putting out something, and maybe we put it in a cage and a bunch of other bugs arrive. That's a behavioral discovery. Okay, the chemistry, the discovery of the pheromone, the synthesis, going through stages 4 and 5. Eventually, we might have made enough in number 7 for testing in the field. That's what a lot of what I'll be showing you are 7, 8, 9. But it takes a long time to figure out how are we going to apply this in both a practical manner that works, but also a responsible manner that doesn't affect potentially non-target species such as natural enemies, parasitoids, and predators, which might also be attracted to these semiochemicals. I'm going to launch into a little bit into my old friend, as Alan referred to. I did my PhD thesis with the Colorado potato beetle. And since that time, Dick Dickens used to be with our group, discovered the male-produced pheromone. And I'm calling it decimdiol. It has 10 carbons, and it is a diol. And the Colorado potato beetle is Leptinotarsa decim lineata, 10-lined. Dick Dickens called it SCPB1, which seemed to me too boring. So I'm going to change that. It's highly attractive to both sexes. We had gram quantities synthesized and are still ongoing with that work. It looks like it might be valuable for trap cropping, but it's not that straightforward. And here's a wrinkle. the female also produces a sex pheromone. We only have behavioral data from that, but here it is. So when you put the along the bottom, if you put out males or females in these cages, and nearby I have a plant which is poisoned with neonic. So when the beetles come, they don't have access to the beetles within the cage, but they come to the adjacent plant and feed on it and drop to the bottom here. So these are the counts. And what happens is when we have the males attracted, are males attracted to both the males and females. You can see that in the top line. Females are only attracted to males. Now there's a wrinkle here in that our colony males aren't putting out as much pheromone. And you have to always be aware of that when you rear insects. You may not rear field quality insects. So in that case, the males from our colony were not vigorous enough, at least in terms of the emission of their pheromone. That was probably a density effect in that we raised them under much higher density than we see in the field. Can you interrupt me if what I'm saying doesn't make sense? Because I got a lot of material here. Please, Matt, do you have a question? No. Okay. We also showed a density, a dose response effect. So if we put out 0, 4, 8, and 12 females in these are just paint strainers that are envelop the plant, very low tech. Paul would call that Walmart science, right, Paul? And we see an effect of the attraction of males. We see no effect. Females are not attracted to the females. So this is a female-produced sex pheromone. So it's very interesting. As far as we know, this is the first beetle for which reciprocal attraction has been shown. There may be some in the audience who could contradict me on that. No? Okay, no contradiction so far. We're going in an interesting direction with this too, because my colleague Jennifer Thaler up at Cornell has been working with the predator of Colorado potato beetle, which is known as the spine soldier bug, Podicus maculoventris. It's a native stink bug, and it's a predator. And one of its favorite prey is Colorado potato beetle. And in Jennifer's lab, they've been working on this for several years. They've shown that the pheromone of the Podicus changes the behavior of the Colorado potato beetle, larvae, and apparently also adults, in such a way that it reduces the populations and causes the larvae not to feed as much, which is pretty amazing to me. So knowing that we had a pull, that is a pheromone, the male-produced aggregation pheromone descendiol, as well as a potential push, which is the predator pheromone, what some of you may know is called fear ecology. We have now a new grant to test whether we could have a push-pull system. The predator pheromone being the push, and the pull being the male-produced aggregation pheromone. So we're very interested to try and put that to work. It's rare. People talk about push-pull systems a lot, but there are not a lot that work. Only a few. My next star is the striped cucumber beetle, and I also show the spotted cucumber beetle, and I'll show you very soon why that's so. The striped cucumber beetle, both are cucurbit pests. They're both native, and the striped cucumber beetle is a very strong specialist on cucurbits and shows up literally overnight when cucurbits, including squash, cucumber, melon, are planted and come up as seedlings, and it's very damaging. So this pheromone that looks a little bit like a I don't know, a dairy cow. Um, that's known as beta-lactone. That was discovered by a person in the audience, Rebecca Smith, uh, about almost 30 years ago now. It's a male-produced aggregation pheromone, and it seems to explain this rapid attack that people see on cucurbit seedlings. The synthesis is difficult, um, but we decided to try an impure synthesis where it results in only producing 8 of the 32 isomers, where only one of the isomers is the actual pheromone. Um, I'm also going to talk, and I want you to know about bitter chemicals called cucurbitacins. They stimulate the feeding of both of these beetles and may be useful in terms of the feeding stimulation to, once they arrive from attraction for the pheromone, to be able to formulate baits which could suppress these critters. The interesting thing is when we put the beta-lactone out and tested it with our clear sticky traps, lo and behold, of course, the striped cucumber beetle was attracted. You can see that on the left, the yellow bars. But lo and behold, the spotted cucumber beetle, which does not produce this pheromone, was strongly attracted. And so were 2 species of squash bug. Um, and you might say, wow, that's really surprising. And it was, honestly. But when you think about this system of this group of vegetables, particularly cucurbita, which is the squash, this is a native crop. All these herbivores we know as pests are native as well. Why shouldn't they be co-evolved and listening or eavesdropping on one another's chemical signals? So really cool stuff, at least for me. Not a surprise here. The striped cucumber beetle responds in a dose-dependent dose response. More means more beetles captured, but so does the spotted cucumber beetle and the horned squash bug. So we have what you might call a semiochemical matrix, where the striped cucumber beetle may be a key, and beta-lactone may be what might be called a keystone semiochemical, or you might look at that beetle as driving the whole process of colonization of a group of herbivores. So it's attractive to spotted cucumber beetle. The western striped cucumber beetle shares that same pheromone, and the 2 squash bugs are attracted. Maybe other Diabotica, same genus as spotted cucumber beetle, we don't know that yet. And maybe parasitoids and predators, we don't know that either. But we do know, due to some recent work in Angel Helms's lab in Texas A&M, that the plant also responds to the pheromone, and changes occur in the cucurbita, the yellow squash they use in this case. And that manuscript's been submitted. Very interesting stuff. The plants are not just passive. They are responding. In this case, again, maybe a surprise, but they're responding to the herbivores' chemical signals.
How are they responding?
How are they responding? So it shows that their resistance to feeding goes up, and also at a molecular level, different genes are being activated and different volatiles are being produced. So there's quite it looks like a complex response. And of course, these, not maybe this specific cultivar, but the ancestors of these did coexist with the Acalema vittatum, the striped cucumber beetle, for millennia before that. So very intriguing. So as I say, we got out and thought, well, what practical value might this have? This is Ariella Haber my former postdoc, and Megan Perlehy-Adams, who's my support scientist, looking at the clear sticky traps. We did not contemplate using yellow sticky traps because they're very attractive to a lot of things, including parasitoids. But we did test them to see how large that effect was. And I don't know if you can see this, but I'm starting out with a clear sticky trap as a standard. And we discovered that the boll weevil trap mounted on the ground was pretty much our best non-sticky trap. And then we had some real losers. And as I mentioned, the yellow sticky trap caught everything. And after a while, that saturates the trap and not much more will stick to it when you got a couple of big grasshoppers and all sorts of Hymenoptera, including pollinators, which we don't want to see. So this was the pattern on the left, what we saw with the clear sticky traps, 2 generations of the striped cucumber beetle, and these are in the borders of a cucurbit planting. And then the dotted line is the proportion of females. So you can see, if you see the first wave, which is in May, is only about it's about a quarter female, so 3/4 male, and they are generally colonizing earlier. And the same thing, a little bit less in the late season when we have the second generation. Then in the second year of this work, we looked at further into April, and also border versus corn versus squash. The corn was adjacent to the squash, and you can see that early season, there's again that big peak. Even before the corn or the squash, anyone would plant that. So we think that the early season is really key if we want to intercept those beetles, which would otherwise move into the direct-seeded cucurbits. We're also working with colleagues at Purdue and Virginia Tech on the combination of floral volatiles. Now, if you know about squash and the fact that they're bee pollinated, that may raise your alarms, and it should. So certain floral volatiles are very attractive to bees, including squash bee, bumblebee, and honeybee, all of which play a role in pollinating cucurbits. But interestingly, one of them, indole, is not. And this is a very good thing because it's very attractive to the cucumber beetles, and it's a lot cheaper than beta-lactone. So if we can get the most out of our pheromone by adding a volatile which does not negatively affect the pollinators and anything else that would be attracted to floral volatiles, then we can combine it with the beta-lactone and really stretch that, what is fairly expensive chemical to make. So we're looking at combinations. If you see the bottom line there, the pheromone, the cucurbitacin, which is the feeding stimulant, the floral volatile, which is still selective, and a fairly benign toxin like spinosyn. Interestingly, cucurbitacins, being bitter, are basically anti-feedants to almost all other insects except for cucurbit specialists. So we can have the prospect to protect the non-targets, including pollinators and natural enemies. So this was one pilot study which Anna Wallingford, my colleague, and I devised along with some entomologists in Vermont and Maine, and this we call roller derby. This is based on a paint roller, which is saturated with bitter watermelon juice. And we tested plus or minus bitter watermelon, plus or minus just plain sweet melon, and plus or minus the pheromone. And they all were important in the attraction of both the striped and the spotted cucumber beetle. So good results there, but a lot of growers are not going to put up a paint roll or a fancy thing like that. Oh, you got to renew it every week. So we really have to look at it and think of it as what is going to work for a small farmer that might be interested in this very low tox, environmentally friendly scheme. I'm going to take a brief return to some of the bugs, some interesting stuff we've been doing lately with leaf-footed bugs. And that's the name of the family Cariidae, but it's also the name of the genus Leptoglossus. And these are becoming more problematic, particularly out west. Leptoglossus zonatus and clypealis are particularly zonatus now is a big problem on pistachios, almonds, and pomegranates. So, and worldwide, there are a number of problematic species. They're all native to the Americas, except for possibly Gonagra, the lower right one. Leptoglossus occidentalis is known as the Western conifer seed bug, and now it's all over Europe and Asia and wreaking havoc with pine nuts, which are a wonderful crop that sustains a lot of otherwise very poor rural pine nut gatherers. If this bug has a great impact on those, some of those pines will not be worth it to keep and maybe cut down. So there's a real environmental concern with that. Getting on to some of the species that we have, Leptoglossus oppositus and Phylopus, the ones on the left there, on the right there, are the ones that we have around in Maryland. And our colleagues at UC Riverside gave us a recently discovered pheromone that's known as leptotriene. It is a triene if you know your chemistry, and it's leptoglossus. So Jocelyn Miller's lab has shared that with us to test on these new species. Also keeping in mind that there's a very important egg parasitoid, Hadronotus pennsylvanicus, recently renamed by Elijah Talamas, used to be Gryon pennsylvanicum, and a very handsome tachinid Trichopoda penipes, which has a fairly broad host range, including both the leaf-footed bugs and stink bugs. This is showing some of the egg parasitoids, which are all which are celionids. This is the Hadronotus pennsylvanicus, and postdoc in our lab, Sean Boyle. This is his picture. It parasitizes Anassa, which are the swash bugs I mentioned earlier, but also really likes leaf-footed bugs and their eggs. And you can see on the left the oviposition of the stick-shaped egg mass. And then here are the nymphs in the middle photo showing the Hadronotus on the right emerging from the egg mass there. They actually prefer Leptoglossus. Oppositus is a minor pest of squash, seemingly becoming more important. And this was just a consideration for the main target, which was the squash bug pictured there on the left. So with chemical ecology, I've had some eureka moments. The main one was with brown marmorated stink bug when we put out the lure known as number 20, and all of a sudden you get 200 in one trap and 2 in another, and you've got something. But the leaf-footed bugs, this is Leptoglossus oppositus here on the binder clip, and they love it. And they don't get too well caught in this sticky trap. I'm not real fond of sticky traps after all this work, but you can see that the 4 bars here that contain leptotriene are very attractive, and the one that isn't. And it's also attractive to the males, the females, and the nymphs. And unfortunately, it's also attractive to Trichopoda penipes, which is pictured here. And if you see on the left-hand side of the sticky trap, there's quite a few there. They're not very well synchronized with Leptoglossus because a lot of them are parasitizing penatomids, stink bugs, and there's some evidence for the fact that there may be some host races there. But this would be a big consideration in any application of that pheromone for anything but just a very, monitoring purposes. If we were to apply it for management of Leptoglossus, we obviously have to keep the Trichopoda in mind and some way to, either with a trap design or some other mechanism to make it selective, even though the pheromone is attractive. That's an attractive tachinid, I would say. Yeah. So some interesting questions come up, which I've alluded to some of them. Um, why are these pheromones attractive to males? Well, the males that are emitting it really don't get to make that decision. There's no obvious advantage to the sender, and maybe a little disadvantage in that emitting pheromone emitting male wants to attract female mates, and maybe not so much the males who might compete with him for them. But that in a way, if you think of the definition of pheromone, where the sender the receiver benefits and the sender does not benefit, we might look on this as what some might call intraspecific pheromone. But there's the advantage, obviously, to the male receiver is that they get to go to where the females are likely to be present, and food is likely to be present as well. So the interesting thing is the fact of whether the male produced pheromone is attracted to just females or to males and females is scattered throughout the genera of stink bugs. So if you look at subfamilies within Pentomidae, the stink bugs, there are some that are sex pheromones and some that are aggregation pheromones, and there are many, many different genera scattered through different families that have one or the other. So I think that question is a very interesting open question. Why are there cross-species attractions? I mentioned some of the ideas of that earlier. Why are these pheromones attractive to the same species of bug nymphs? And actually, Dick Dickens showed that they were actually attractive to the larvae of Colorado potato beetle, the male-produced aggregation pheromone. There are some ideas about that. One is that obviously, if there are adults putting out pheromone, there's food. And the tests that have been done with the nymphs and larvae have been starved or at least away from food. So they may be using that just to find food. Usually the nymphs are happy. If they're happy on what food they're on, they're going to stay put. However, we found, and Tracy's lab has confirmed this, especially for brown marmorated stink bug, they can move many meters once they become pretty old nymphs. The aggregation with adults, and this is true of the bugs at least, may improve food quality. You know, it's very well known for aphids, but it's also known for some true bugs. That once a group of bugs is feeding on a plant, it basically coerces the plant and to send nutrients to where the feeding site is, or what might be called a resource sink. And also, it's been proposed that the immature insects are attracted because of future mating opportunities. However, if you remember back to the brown marmorated stink bug diagram by instar, there was no difference. You would expect that if that was a strong effect, that would be nymphs that were about to molt and be adults and want to mate, and that we didn't see. Finally, we're lucky about the fact that, excuse me, there are two more things. Why is there no apparent upper limit to their attractiveness? That's kind of an interesting question, and it's beneficial from a management standpoint. In nature, there's evidence for self-regulation. So if there are a bunch of males together, they throttle back on their pheromone production. Why would they do that? Well, maybe it's expensive to produce or to release, so they're going to let the other males do the job. If you think about if there's 10 males there, there's also likely in nature, at least if there's a 1-to-1 sex ratio, for there to be already some females there. So it's kind of an unusual situation that you would have 10 males and no females, for instance. So why should they continue to produce pheromone? They should look for the local females. So the other idea that is just that why is there no upper limit? The females may be responding to what might be looked at as an olfactory peacock effect, sexual selection, where if a male can produce a lot, that is considered a very highly qualified mate. It's a proxy for a vigorous male, such as has been repeatedly hypothesized for peacocks, for instance. Yeah.
Has that been compared between, in the case of the brown marmorated stink bug, has that been compared in its non-native range with its native range? Because it
Not,
seems like they've gone through a weird bottleneck, at least in North America.
not enough.
To throw all this off.
So Rob Morrison had some work over in Korea, and it wasn't detailed enough to see that. So there wasn't a set of doses, but also what I was interested in, would the Plautia stali be attracted? Was there a reciprocal attraction? But the Korean cooperators didn't count the Plautia stali. And they also didn't do different doses. So it's a good question. It's a good question of whether that would work. However, that's true with harlequin bug in terms of the no upper limit. That's true of some of the other species I'm talking about, including harlequin bug. Now, another benefit which is useful from a management standpoint is the presence of some of the off isomers that I showed you that are present in both the commercial preparation of brown marmorated, which is also used for harlequin bug, and in the V-tadalactone mixture we use. It doesn't seem to be important. If you're familiar with the Lepidoptera pheromone work, it's often absolutely critical to have the right ratio. And if you have too much of the other ratio or too little, you're done. It doesn't work. They're very sensitive to that. Keeping in mind now, those are much smaller molecules and probably used by multiple species. So maybe that's why the ratios are important. Whereas these complex molecules, many of which are, big terpenes, maybe present more of a unique chemical signature where the bugs or the beetles that are responding to that without regard to the exact ratio, because it's such a rare thing to be out there anyway. So who knows? They may be less sensitive, at least in the case of the true bugs, because they're using it for distance attraction. They go and see what's there. And actually, it's been demonstrated, and this may be also true for Colorado potato beetle with a female produced pheromone, that once the distance attraction is achieved, once they're in the same area, for instance, with bugs, there's specific courtship that's mediated by vibration through the plant, visual effects, and as I mentioned, maybe even female produced pheromones. So, bunch of interesting Evo stuff. From a practical standpoint, these are all the sorts of things you can think about. Certainly a lot are applied already for monitoring with traps, or in some cases trap plants. We're looking at mass trapping. Traps are expensive, so maybe it's better to look at supercharged trap crops, that is, trap crops that are supplemented with pheromones and other attractants. I mentioned the push-pull systems. And in Japan, they've done some work with pheromone-mediated auto-dissemination of entomopathogenic fungi. And also another thing that is kind of intriguing is using the aggregation pheromones of some of the predatory insects, such as Podicis, to manipulate biocontrol in the field, but also the eavesdropped chemicals, so to speak, that tachinids and some Hymenopterous parasitoids use in order to perhaps concentrate biocontrol where it's most needed. So those are just some kind of ideas that all the references at the bottom have at least tested experimentally. And then some, as I mentioned, a lot of hurdles on the way to do that before you really develop a system and have impact. And we've got to keep in mind that, just having the knowledge of the chemistry is our knowledge of the biology really needs to match up to that before we can apply. And keeping in mind multiple functions of pheromones, and like I mentioned, the other senses involved, and the fact that particularly the bugs, but also the beetles, species you need to know about the wild host and how it functions in the agroecosystem. Um, Here are some maybe crazier few or further away prospects for novel applications of semiochemicals. Um, Work a bit with Dorothea Toll at Virginia Tech on plant biosynthesis of some of these terpenes. They make, in some cases, they make the same chemicals. So could we, for instance, breed a trap plant, which was just for that purpose, is a possibility. That's actually happened with a few of the simpler left pheromones. Um, could we use this for remote olfactory sensing, mobile autonomous trapping vehicles called Pied Pipers or whatever you might call them? And as I mentioned, manipulation of natural enemies in the field. But any of these requires a really careful evaluation of each system. I mentioned the interaction of the pheromone, potential use of pheromones for, say, attract and kill, versus a consideration of what natural enemies are out there, how are they responding? If they respond, how are we going to make the system selective so that it doesn't negatively affect that? And makes it so that all tactics, including biological and the behavioral controls, can work together, which is kind of the essence of integrated pest management, or IPM. So I'd like to open the floor for questions, but first to thank my many cooperators on the number of studies I've shown here, both at universities and ARS, and some of you in the audience, I'm sure. Thanks. Want to keep there's an extra couple slides, so keep that going. That's a black slide.
Oh, well then.
If anybody has any questions about flea beetles, for instance, which I didn't show, even though I showed it in my introductory. Go ahead, shoot. Yeah.
How are plant traps deployed, and how exactly are they effective?
So I showed one experimental use where basically you have a poison plant and then you have an attractant associated with that. You can deploy plants with pheromones or other attractants as trap crops. There's always the issue of, okay, I got an aggregation on my trap crop. Over here's somewhere is a main crop I'm trying to protect. And now what do I do? If you're really lucky, maybe the insects stay on your trap crop. But as I mentioned, there's this whole dynamic of attraction and arrestment, which can fall apart, especially once you get high densities on your trap crop. So it's a good question, and one that you really have to deal with in terms of trap cropping. The easiest answer is attract them there and kill them somehow. It may be through baits, which aren't even plants, or it may be through plants which are inherently dead end hosts. There's some work that's been done in Europe on co-crops with that. So there's a lot of different avenues there, but always the awareness that you create an aggregation and you create a potential problem too. Some other questions? Yeah.
Have you tried using isothiocyanates to attract some of the chrysofeliders?
Yes, absolutely. Buried within some of my slides there for harlequin bug was the fact that they are quite attractive. I didn't have the best slide, which shows that allyl isothiocyanate, which is a very cheap and actually somewhat toxic, but it's also in wasabi, so the dose is the poison, right? But that's very attractive, especially in combination with the harlequin bug pheromone. Now, some other crucifer specialists don't use the isothiocyanates. But I'm glad you asked that because I think I have an extra slide there. Okay, let's see. If I, oh yeah, you're gonna let me, okay. So at the very end here, I have some flea beetle work that shows that they're the crucifer, oh, I guess I don't have that. All right, the crucifer flea beetles show a very strong attraction to allyl isothiocyanate, and in combination with their known aggregation pheromones, it's even stronger. So them and the harlequin bug, it's a good example. And from a practical standpoint, the plant chemicals are usually available off the shelf and they're fairly inexpensive. And so you can, again, use them to stretch the effect of the typically much more complicated and expensive to make pheromone. That's a good question, and I overlooked it in my presentation.
I tried using isothiocyanates for flea beetles overseas, and it was moderately successful for Philotreta cruciferi.
Okay, so by themselves, we got variable results, and they varied by region. We had a cooperator in North Dakota, and the results from there and in Virginia and Maryland were different. So it suggests something's going on there, and I don't know quite what. And that was true for the striped cucumber beetle too. And we published that with Alex, and still leaves open the question.
Cruciferi is not native here, actually; it's introduced.
Well, the specific species of cole crops and canola, where they're big pests, are introduced, but there are native crucifers and there are native flea beetles that go to those crucifers. And in fact, with the isothiocyanate, we got some of them. Now, it wasn't very much, and it's out in the middle of the field, and their native host is, in this case, Dentaria lacinosa, the native toothwort, which is a spring ephemeral. So probably the synchrony threw off any captures we would have made. But it's a very interesting group, and unfortunately, Miklos Tot in Hungary, who did a lot of work with them, has retired now. So we need some European work on that. Questions from okay.
Not a question important to the system per se, just something about the basic biology. Are these species typically monandrous, or do they make multiple times with multiple different males, or is one good enough?
As far as is known, for Colorado potato beetle, it's quite well known that they mate multiple times. It appears that striped cucumber beetle, that's true as well. I believe that's also true for the stink bugs, but I'm not as familiar with that. But I think it's typical. Yeah. Questions from the online audience?
I have a question. I'm curious about the host plant preferences of Leptoglossus. It seems like they're a little bit varied. Some of them being on conifers and others being, on things like pistachios and whatnot. How well studied is Leptoglossus in terms of like its phylogeny and the evolution of host plant preferences in that group?
Oh my gosh, I wish somebody would do it. There are about 60 to 70 species. They're all apparently native to the Americas except for Leptoglossus gonagra, which may or may not be an early invader of the old world. You're right, the host preferences are kind of all over the place. There's some very, very wide host ranges, and in terms of the occidentalis, they're very tight on the conifers, not just the pinus, but a few related conifer genera. It would be a great study. I believe the author who's in Mexico City, Balachowski, has done very good work on the taxonomy. So I think what hasn't been done is both the molecular work that would help determine the phylogeny, and also, as you mentioned, the relationship of that to the host preferences. I mean, bugs in general, and this is true of, say, Zonitis out there, they love pomegranate, and they build up on that because it's not a very managed crop in the Central Valley, and then they go to pistachio and almond, which are in completely different families. A lot of the bug preferences have to do with what plant part it is and what stage it's at. They often attack immature fruits or flower buds. So I mean, if anybody's interested in doing that, it's kind of a wide open field. I didn't mention, but Phylopus, the other species that's found around here and a big problem in the south, doesn't respond at all to leptotriene. So that's another mystery. Like, what is its pheromone? Who knows?
I'm a bit surprised that nobody's really looked at that, considering one, that it's a massive pest, two, that it's such a large and charismatic insect, and three, that it's so incredibly diverse. I mean, it's got, we're talking about a genus with 60 some odd species.
I completely agree, but sometimes our biodiversity overwhelms us, doesn't it?
True.
Any other questions?
Ron, it's a really interesting talk. Quick question. You mentioned in the middle of your talk, you mentioned the plant can actually sense the bug pheromone, induce the plant to produce resistance.
That's right. That was demonstrated first in goldenrod in response to the gall producing fly, and I can't remember the species.
Wow.
Yeah. And so that was demonstrated there. This may be the first demonstration in agricultural crop, and it's not yet published, so I don't want to go into all the details, but it's a definite response, and it's it involves essentially upregulating herbivore resistance to herbivores. Yeah, it's kind of exciting.
Oh, it's really interesting and exciting. Sorry, I couldn't be at the site. I wish I'll come maybe next time.
Sounds good. Join up. All right, thanks everybody.
Right. Well, thank you very much, Don. That was an excellent and very interesting presentation. So we have come to that point in the evening. Are there any other articles of business or of note, generally speaking, that anyone would like to bring up at this point? I'd be happy to open the floor. Hearing none, I will now adjourn the 1249th meeting of the Entomological Society of Washington. Thank you all for coming. Have a good evening.
Bye, Andrew.
Bye, Andrew.
Thank you,
Thanks.
everybody.
Bye, everyone.
Toodles.
Good news.
Tomorrow, next month,